Hello everyone, my name is Sage Wackett. I will be presenting on the comparative analysis portion of the complete genome sequence of the Microbacterium bacteriophage Erla, which was a study done at Arizona State University as part of a course program under the CPhages HHMI program. And this manuscript was published by the American Society for Microbiology in January 2021. So for the investigation of bacteriophage Erla, it was actually found at the University of Ottawa, Canada in a garden. And then it was sent to the Pittsburgh Bacteriophage Institute to be sequenced. And then that sequence was sent to us at Arizona State University, where we then annotated it and analyzed it. And you can see from this electron microscope image that bacteriophage Erla kind of shows this cyphoviridate morphology that we can see an example of, a little bit more of a cartoonish example here with this long tail tube, which is very notable. As for the annotation itself, bacteriophage Erla is in the EA1 subcluster and so infects Microbacterium foliorum. And we were given 63 predicted protein coding genes, but Daniel Sacker will actually talk about and discuss why it is we actually came up with a different number. So there's a very interesting discovery that we made there. So I suggest you check that out. So as for the, I guess, how this comparative analysis was undergone, first we took a blast analysis wherein we took the genome of bacteriophage Erla and aligned it to other bacteriophage genomes in the subcluster. And the top three hits here you can identify are Calyx, Gello, and Etta in order. And you can see here their percent identities are very, very similar, very, very distinct from the rest of these bacteriophages. And so really what their difference is this total score here, which identifies how good the alignment is between these two genomes. And we can see here that Calyx has a marginally greater total score, so it is the best alignment. So this Caline analysis kind of puts it into perspective even further, wherein we have Gello, Etta, and Calyx, and Erla in the middle here with 100%. We can identify that Calyx, Etta, and Gello are the three top best alignments in this matrix here. And we can see that Baines and Nattles, which are also in the subcluster, are definitely further off. And we can also see in this visual representation of this phylogenetic tree here, we can see that Calyx, Gello, and Etta are very close to Erla, while the rest are kind of further off in this other group here. So we also used a program called Famirator to essentially take these bacteriophage subclusters called FAMs to essentially compare our bacteriophage to other bacteriophages in the subcluster visually. So you can see here, each of these boxes represents a different protein coding gene. So for instance, number 2 is terminase in Erla, and you can see in Calyx as well, terminase. So this is the basis that was worked on to essentially take each one of these lines and compare them to one another. And using the human eye, we can see that there is a distinct pattern amongst all of these bacteriophages in particular, which reinforces this thought that these three bacteriophages are mostly related to Erla, boxed here in red. So to summarize, we have a narrow range of similar phages that I've just given to you, these top three, Calyx, Gello, and Etta. And after narrowing these down, we can really, in the lab, perform more comparison for their virulence. We can have determination for alternate hosts to essentially provide greater efficacy for phage therapy or other products for bacteriophage manipulation. So I would like to conclude by thanking both doctors Suzanne Pfeiffer and John McCutcheon for spearheading this project, as it wouldn't have been possible without them. I would like to thank Arizona State University and the CPhages HHMI program for really allowing us to undertake this project, myself and other students. And I would like to also thank the American Society for Microbiology again for publishing our manuscript. Thank you all for attending.